Welcome, everyone. My name's Jay Scott. I'm co-executive director at Alex's Lemonade Stand Foundation. I want to thank you for joining us today for this virtual lecture. We have an exciting lecture today. I think this is the 9th lecture in our series. Next week, we have 2 machine learning lectures. And the week after that, we have Charles Mulligan, who is giving another leukemia lecture called The Many Genomic Roads to Acute Lymphoblastic Leukemia. Just some housekeeping. If you have any questions, please type them in the chat. If you have suggestions for other lecture topics, please put them in the survey posted to the chat. And I'm really excited to tell you that today we have a lecture called On the Way to Pediatric Leukemia, Following Clonal Dynamics with Cellular Barcoding, presented by Dr. Leonard I. Dr. I is the Grousbeck Professor of Medicine at Harvard Medical School. He's also a Howard Hughes Medical Institute investigator and the director of the stem cell program at Boston Children's. Dr. Zahn is internationally recognized for his pioneering work in stem cell biology and cancer genetics. He's done a lot of work in zebrafish. He is the founder and former president of the International Society of Stem Cell Research, and he has many, many other honors and awards. But I'm really excited to have him here today to give this lecture. And with that, I will turn it over to you, Dr. Zahn.
Great. Well, thanks, Jay. Let me just set this up. Can everybody see my slides?
Yes.
Great. Okay. All right. Well, I just want to do my disclosures quickly, as I'm a founder and on the SAB of 4 companies, Fate Therapeutics, Scholar Rock, Camp4, and Amagma. And first of all, I want to thank Alex Lemonade Stand Foundation and also the RUNX1 Research Program for funding part of this work. These 2 organizations have really done a lot for understanding pediatric tumors, and I'm very thankful. I should also say I'm thankful for Alex Lemonade Stand for funding my daughter in 2014 when she was a summer student in Ben Ebert's lab, and she just started her oncology fellowship last Wednesday at Dana-Farber, so we're very excited for her. So I wanted to tackle the origin of pediatric leukemia question. If you think about the fetal liver, that is the major site of blood development during fetal life. And it's been thought for many years that fetal hematopoietic stem cells are selectively removed in some way after birth. And these are replaced by adult hematopoietic stem cells, which will drive blood formation in adulthood. But one of the theories of leukemia, particularly pediatric leukemia, has been that there's a fetal progenitor or a stem cell that gets transformed, and this causes an expansion of blood cell populations, and this sets up a preleukemic state. And on top of that, there are other mutations that ultimately lead to leukemia. And although this has been a popular theory, it's never actually been proven. And I believe that some of the technology that I'll talk about using cellular barcoding may actually be able to lead to an understanding of which cell started the pediatric leukemia and also when that occurred during development. And I think this cellular barcoding is very exciting as a new topic. So what is cellular barcoding? Well, I put here a supermarket example on the top. You start with a barcode, and every item in the supermarket has a certain barcode. And you can use a scanner and basically determine what the barcode represents. And that allows the person at the checkout to basically identify a particular food item. It knows the food item and it knows where it came from. And then ultimately we pay for the food so we can taste it and see that it's the right item. Now, in cellular barcoding, what's happened is every cell is actually barcoded with a DNA sequence. And typically we use CRISPR for this approach or some type of recombination technology. The scanning mechanism is to amplify a particular DNA and to sequence the barcode. This will ultimately tell you the history of the cell. It will tell you where it came from, what the cell is, And then ultimately, we look at RNA expression as what the cell is executing, what's its function. Is it a red blood cell, a white blood cell, a platelet, etc. And this technology has really dramatically increased over the past 3 years. And we're very excited in the lab to apply this to a pediatric leukemia approach. So on this slide are a variety of different techniques for cellular barcoding. I'm going to spend a fair amount of time on ZebraBow, which is a technology that allows every stem cell to be barcoded in a different color. I'm also going to talk a lot about Gestalt, which is using a CRISPR barcoding system. Fernando Camargo's lab has pioneered the use of transposons to be able to individually jump transposons in the genome to give every cell a particular code. He's recently developed a CRISPR technology called Carlin that also reads out as a single cell RNA expression. And then in humans, it's possible to use recombination that happens during mitosis as a method to barcode cells. And also, it's possible to use mitochondria, as Vijay Sankaran has shown us, to use these barcoding systems. And all of these are incredibly useful. And what we're seeing is the ability to compare things between fish experiments, mouse experiments, and human experiments is quite powerful. So what we can think of if we're thinking about leukemia, in a typical patient, let's say an adult patient, during development, there's a diversity of stem cells. And I've just put here a number of different colors because every stem cell has a different approach. But what happens as people age is a syndrome called CHIP, or clonal hematopoiesis of indeterminate potential. In this particular situation, mutations happen in the blood cells, and this causes a clonal dominance. And this could ultimately lead to a myelodysplastic situation, or ultimately with more mutations to a leukemia. It's interesting that there are really no symptoms of having this expanded state of cells. And I think this is very relevant to what we're seeing in pediatric leukemia as well as adulthood. So as many of you know, I use the zebrafish as a system to study blood cells. And I'm just going to give you a little movie to show you and orient you of the birth of the blood stem cells during fetal or larval state. So here we have the dorsal aorta. And what we're going to see is a blood vessel actually changes its fate and it rounds up to form a blood stem cell. That stem cell then goes off into circulation and is going to colonize the equivalent of the fetal liver, which in the fish is in its tail in this caudal hematopoietic tissue. Now this cell will actually land in that particular area and try to set up home. It will actually transmigrate through the blood vessels where these cells will actually double, and then cells will go back into circulation and land in the marrow. And for us in the fish, the kidney is the site where the marrow is. In humans, it would be in your bones. And this sets up the kidney as a hematopoietic organ. Some of the cells bypass the kidney and go to the thymus to start the immune system. And all of this lets the animal have blood really throughout its entire lifetime. So several years ago, John Henninger developed the Rainbow system for examining blood cells. And what he did was he wanted to know just how many blood cells were made during embryogenesis. And so he had these rainbow fish, which have 3 colors, red, blue, and green. And they're separated by recombination sites. And there's about 15 to 20 copies throughout the entire genome of this fish. When he mated this to a fish that has an inducible Cree, which is blood specific, he could turn all of the embryonic blood a different color. Now, most of the embryonic blood is removed from circulation as the adulthood happens. And he could look at the colors in these adults, and whatever colors he found, he could back calculate how many stem cells were actually made during embryogenesis. And this is a movie of the blood cells of an embryo, and you can see they're multicolored. And if you focus on this particular cell here, it's executing a certain behavior that makes me think that this is a blood stem cell. Now, this blue-green cell, when it grows up to adulthood, will actually make blue-green blood. So now we can grow the animal up for 3 months, 4 months, and see what happens. And this is the peripheral blood of that animal. And you can see there's many different colors represented. So using this, he could start counting those colors. And what he found is that if he labels the cells at 24 hours with the inducible CRE, he could find 21 clones of stem cells are the ones that are being born. And the number increases as time goes on as you head towards that fetal liver equivalent stage. So this shows that the barcoding method of making cells different colors actually works very well. And we wanted to model this now to the transformative events that happen as clonal disorders occur. So in humans, you have polyclonal hematopoiesis, multicolored, but you can acquire mutations, and then these mutations will lead to clonal expansion. In the fish, what we have are the ability to change every one of these stem cells into a different color. And then we can target either transgenes or CRISPR and inactivate some of the interesting genes that will ultimately lead to clonal expansion, and thereby modeling the human disease. So we invented this technique called twister. I hope everybody will appreciate the twister like the game. But the idea is it's a way of using tissue editing with inducible stem cell tagging via recombination. The concept is here, we'll take guide RNAs to 20 genes that are pretty well known to cause this clonal expansion in aging populations. We will then do this in the background of the colored fish, and we will induce the colors just before the birth of the stem cells. This way, we will be able to see if there's any type of clonal expansion that ultimately happens. And what's nice about the colors is that you can actually sort the dominant clones and use analysis to DNA or RNA-seq to study what those genes are doing. So what we saw here was under controls, you don't see much clonal expansion. In fact, most of these here are more likely that there's two colors that are pretty close to each other that just aren't so resolvable. But here, with injecting these 20 guides that target roughly 20 genes, you can see you get clonal expansion. And you can see that, for instance, this animal, 70% of the blood comes from a single color, a single stem cell. Now, when we look at the types of mutations that occur, we can see that this is a very similar distribution to what you would see in the human aged population. You see mutations, for instance, in ASXL1 that are pretty well represented. We average about 5 or 6 mutations per fish. And this allows us to rapidly screen through things that would look for, again, clonal expansion. Now, the nice part about this is the fluorescently labeled clones can be sorted. So for instance, in this case, Serena Vagian, the postdoc who did this excellent work, along with John Henninger, a graduate student, you can see we can sort this population, and we know that the mutations that occur in each individual color are distinct. And then you can also sort a non-dominant clone, and you can tell what the populations are based on their mutations. What we see is that when we've targeted particular types of genes, that from 3 months to 7 months, there ends up being a clonal expansion. And again, we think that this faithfully reproduces what is a human clonal expansion. And this 7-month animal would be getting towards the aged population. So then once you have this, you can line up all the mutations, and each one of these columns here is a fish, a single fish. And it has, for instance, an ASXL1 mutation and a P53 mutation, a DNMT8, which is a DNMT3A mutation. And you can see here by far that in the dominant clusters, ASXL1 gives you a dominant clone. Now, the mutations in ASXL1 are all in exon 12 that cause this. If you knock out ASXL1, you don't get this type of dominance. And this is true also in humans. Now, many of the mutations are represented in both the dominant and the non-dominant clusters. And this tells you that the clonal expansion occurs in each of these individual populations by a variety of different mechanisms. Now, one of the nice things is we've been able to sort those cells and then look at RNA-seq. And when we do this, we see that in the mature lineages, let's say we look at IL-1 or TNF-alpha, we see that this is higher in the macrophages than the neutrophils, but is also true a little bit in the stem cells. And if you look at these populations, the IL-1 and TNF-alpha are thought to drive some of the clotting abnormalities that are seen in these patients who have clonal disorders. One of the things that we've been able to do is to look at the actual stem cells themselves. And we saw here these anti-inflammatory pathways, NR4A1 and SOX3, and these are upregulated in the stem cells. So what's interesting is we think the mature lineage cells are producing inflammatory markers, and the stem cells are producing markers that are resistant to those. And that might actually be a mechanism for clonal expansion to actually occur. So another way of saying it is that the inflammation is driving some of this competitiveness to allow a clone to expand. These mature myeloid cells are secreting IL-1 and TNF-alpha, but the stem cells that are surviving are actually resistant to this. And so this allows for a selective type of process. Now, what about pediatric leukemias? So we've begun to look at cancer predisposition syndromes. And in particular, the work I'm going to tell you about is on RUNX1 and GATA2 mutations, which have been linked to pediatric leukemia. Now, in the fish, we've been able to, with Paul Liu's group, examine the RUNX1 mutant zebrafish. Now, interestingly, RUNX1 mutant fish actually live to adulthood. And what they have is a deficit of myelomonocytes, and this is shown over here. Now, when we do this in the background of the clonal marking, what you can see is the wild type and the heterozygote doesn't have much of a change of all these colors. I would say typically we have about 20 colors. But you can see in the RUNX1 knockout, there's a deficit of colors. And this would suggest that these adults that have gone through probably a bottleneck of stem cells to survive, they actually have fewer clones. And we think this is very relevant to the pediatric situation where people would have RUNX1 mutations. Shown here is just a look at the cluster size. And you can see here the higher cluster size of the knockout, meaning the individual clones are larger with respect to the RUNX1 knockout. And then we looked at a GATA2 deficient animal, and Serene actually made a GATA2 mutant for the fish. And this fish has myelomonocytopenia, which is very similar to what happens in the human situation with GATA2 mutations. And you can see there's an overrepresentative progenitors compared to mature lineages. Now, the surprise here, again, is when we look at just the GATA2 heterozygote, we get single fish that already have clonal hematopoiesis. In other words, these fish are already have an expanded pool. And you can see here the percent of fish with a dominant clone, and the heterozygote leads to a larger clone size. So we're now looking for secondary mutations that would stimulate GATA2 deficiency to transition to AML and to also cause this expanded population. And that's work that we're currently doing. So in the wild type situation, you're born with a diversity of stem cells. And these stem cells will go on to ultimately populate the adult marrow, and there's a diversity of stem cell populations. With CHIP of aging, you have a mutation that occurs during elderly development, and that mutation leads to an expanded clone. And then mutations that further happen can lead ultimately to the malignancy. In the pediatric situation or congenital situation for RUNX1 or for GATA2 deficiency, you're born with a mutation. This leads to clonal dominance, but the mechanism of clonal dominance is actually due to a decreased number of stem cells or the effectiveness of those stem cells to interact with the niche. But the output at the end looks very, very similar with this clonal expansion happening. So you can get leukemia either by having an expanded pool based on mutation later in life, or by having a dearth of populations, as is true in the congenital situation. Now I'd like to talk to you about Gestalt, which is another barcoding system. And actually, I've never talked about this work really before. So I'm very excited to give this part of the lecture. So Gestalt stands for single cell genome editing of synthetic targeted arrays for lineage tracing. It uses a CRISPR barcode. I'll explain this in a second. You can establish lineage trees based on this approach. This technology in 2018 won the breakthrough of the year, and it's very exciting that we can now, just a couple years later, apply it to clonal expansion. So Gestalt has two different fish. So on one of the fish, there's a barcoding fish. There's actually 10 positions for a CRISPR barcode. To be fair, we don't use this 10th position, so we use the first 9. Then we have a guide fish. This is a fish that has the ability to actually make the guide that would target these particular sites in the barcoded fish. And this is inducible with Cas9 through a heat shock mechanism. So the elegance of this system is at the 1-cell stage, we can inject 4 guide RNAs and target the first 4 positions. This allows us to label all of embryogenesis with a certain barcode. Then with a heat shock mechanism shown here, we can come in and barcode the rest of those positions. This allows us to have another stage. For us, it'll be just before the birth of blood stem cells, when we're going to label the sites 5 through 9. Again, this allows us to ultimately tell you when the expansion is occurring of a particular clone. So far, and this is early data, we've analyzed 9 fish so far. And you can see that we have a variety of different methods. One is a heat shock method, and one is using a ubiquitous promoter. It turns out that with the heat shock method, you don't get quite as many barcodes as you do as having a very strong ubiquitous promoter. And so we would probably think that this approach is the more preferred one. Now we can examine the marrow, but we can also examine the thymus and look at the relationship of the marrow to the thymus. And you can see we get substantial numbers of barcodes in any case, which is very, very exciting. So with this particular technology, the barcode is read out as an RNA. So the first step is to do single cell RNA-seq. And what you can see here is 71,000 cells of the marrow being analyzed by RNA-seq. And this looks very, very similar to what you would see, let's say, from a human marrow sample. And all the different blood lineages are represented. Now, if you think about this, we have all the cells that I just showed you for which there is RNA-seq data. But then a subset of those cells will actually have Gestalt barcode information. And it's the intersection of looking at the transcriptome information as well as the barcode information that allows you to say something about a particular clone. You can see this is just one clone that we highlighted for you, which is present in the myeloid cells as well as really all of the blood cells. So this is my favorite video. And what this represents for this lecture is all of the individual clones that we're able to read out. And you can see sometimes a clone is more myeloid specific, sometimes it's lymphoid specific. And it's very, very exciting to see the resolution that we're getting from this Gestalt barcoding. So what we have are different types of clones are now identifiable. So for instance, over here is a clone that represents panhematopoiesis. It's an unbiased clone. So all the blood cells are actually read out. Over here is a myeloid biased clone, and you can see predominantly granulocytes and monocytes being read out. And over here is a lymphoid biased clone where there's hardly any myeloid cells being read out, but all the lymphocytes are actually being tagged. Now, one of the first surprises that we found was, again, we're labeling during the equivalent of fetal life and following these clones all the way to adulthood, maybe 3 or 4 months later. And what you can see here is that some of the clones are myeloid biased and some of them are lymphoid biased, and then others are balanced. And these clones, though, that are tissue specific or lineage specific are persisting from fetal life. And that is a real surprise. And this persistence happens for a very long time because we've even analyzed some fish that are 6 months old. And again, that's getting to be pretty old as a fish. So again, this makes you wonder if these types of cells aren't the origin of the pediatric leukemias. So if we look here at a systematic clone analysis, this is just for 5 of the fish. And I'm just going to focus, let's say, on clone 48, you can see that many of the clones are unbiased that come from clone 48, but there is a subset that's lymphoid biased, I should say, and there's also a subset that is myeloid biased. And so I think our data shows that there are long-lived fetal progenitors that persist into adulthood. And again, I would suggest that this is the population that ultimately is leading to pediatric leukemia. Now, the other thing is we can start taking apart individual lineages and seeing, are there specific clones that go to particular lineages? And so these are all the peripheral blood lineages. And you can see that we are seeing very specific clones. Again, each of these are the clone number, but you can see, you know, things that are only granulocytes or only lymphocytes or NK cells here, you know, so it's a very interesting population. And this just shows some of the clones that we thought were interesting. For instance, here is one where they're only making platelets, thrombocytes. Here's another one which is only making granulocytes. And here's another one that's certainly erythrocyte biased. Now, Sarah Rubin and Chloe Barron, who have been doing the majority of this work, Sarah was particularly interested in the thymus and what is the relation of the thymus and the marrow. And so from the same fish, she was able to capture the marrow and the thymus with barcoded cells. And here we got a second surprise. And that second surprise is there are thymic T cell specific clones that are not represented in the marrow. Okay. And that is also surprising. And I asked a bunch of my friends who are immunologists what the result should be. Should all of the clones in the thymus be represented by the marrow? And there was a 50-50 split in terms of opinions. So I think our data actually answers a fundamental question, that there are clones from fetal life that just generate the lymphocytes, and they are lymphocyte specific clones. So the Gestalt system allows for embryonic barcoding and looking at the stem cell birth and following these cells throughout life. Barcoding at 28 hours post-fertilization allows you to look at lineage and looks at bias of individual stem and progenitor cells in the adult marrow. And then with respect to the T cells and the lymphocytes in general, I think it's surprising that there are long-lived lymphoid progenitors that occur in adulthood that come from fetal life. And a second surprise is that there are thymus-specific clones that aren't represented in the marrow. We're now generating lineage trees. We hope to have this done pretty shortly. And this is also very exciting because it will allow you to trace back during embryogenesis and see when particularly was that transformation event happening. And that's going to be another exciting set of experiments. Now, with regard to this type of lineage barcoding, Fernando Camargo's lab has been leading the way in many ways with regard to barcoding. And this is the work from the Camargo lab, where he's used a fate mapping type of technique, FLT3 locus, using an inducible Cre-RT2. He finds that hematopoietic progenitors are long-lived. And he also finds in months post-birth that there are long-lived lymphoid progenitors as well as myeloid progenitors, very similar to what we see in the fish. And here he's mapped, and you can see some of these cells start decaying, 9 months, even up to a year afterwards. And so I think what we're seeing in the fish and what we're seeing in the mouse are very, very similar. Now I'd like to finish with a story about, well, it's great if you can use clonal marking to look at the lineage, but what could you do about this? Or is there anything therapeutic you could do to modulate individual clonal populations? And this is the work of Sam Watrous, a graduate student in the lab, who has some very interesting data around the interaction of macrophages with stem cells and how they may modulate clonality. So Sam was studying that fetal liver equivalent niche, and he had, along with Elliot Hagedorn, shown that these red macrophages would actually get very, very close to these stem cells and what we call groomed them. It would actually remove cytoplasmic material. You can see that green is moving. And this seemed to be a very intimate interaction, and we wondered what its purpose was. And so Sam went ahead and did some more imaging. Now our stem cells are marked in M cherry in purple. The macrophages here are green. And we saw some crazy behaviors. So I'm going to try to play this movie. So the first behavior we saw was a stem cell that was completely eaten by a macrophage. Again, this was in the fetal environment. This was surprising, and about 20% of all of the stem cells are selectively removed from by the macrophages. Then he saw a second behavior where the macrophage and stem cell interacted, but instead of fully eating the stem cell, it actually just partially ate the stem cell, and then the stem cell divided. And it's almost as if the macrophage interacting with the stem cell is a licensing event for the stem cell to actually divide. So this raised a number of questions. What are the frequency of these events? What determines this interaction? Is there anything special about these macrophages? What determines being doomed or groomed? And what is being monitored on the stem cells? So it turns out that about a quarter of all the stem cells are actually interacting with a macrophage in this fetal environment. And this is true across different time periods. We also know that it's relatively specific for stem cells. So for instance, if we look at red blood cell interactions with macrophages, that hardly happens at all, but the stem cells definitely interact specifically. So we have a couple techniques where we're able to remove the macrophages. One of the techniques is to feed the fish clonal liposomes. This is eaten by the macrophages and kills the macrophage. The other is to genetically ablate the macrophages by using an IRF8 morpholino. This genetically is required for macrophage development. So when we deplete the macrophages, we don't see much change in the overall number of stem cells in the body of the fish. But when we use our color barcoding, we see something quite remarkable. So again, here we used the colors, we gave clonal, and then we looked at stem cell number. It turns out if you delete the macrophages, you get half the number of stem cell clones compared to a sibling control. And you can see here, again, it's normally we have about 25 colors. With clonal, you reduce it to about half. If you use the IRF8 morpholino, you also reduce it to half. And even if you give the clonal later, well past the birth of the stem cells, you have this clonal dominance here as a result of probably having deficit of divisions of stem cells. Now, the macrophages that have eaten the stem cells, because they have a little bit of red stem cell in them, we can fax purify the macrophages that have eaten the stem cells. And then we did RNA-seq. And what we saw were pathways that were very involved in engulfment. And in particular, there were a lot of lipids, of apoproteins, that seemed to be being in those macrophages that were eating the stem cells. So we used a dye called cholesterol bodipping. And the hypothesis here is that we wanted to see, are the macrophages that are eating the stem cells also going to be taking up cholesterol? And so shown here, if you look over here, as the stem cell arrives, you can see that the Bodippy is being taken up by the macrophage that has eaten the stem cell. And when we look at this, it's very statistically significant. The engaged macrophages have taken up the Bodippy, whereas the unengaged macrophages have not. So we wanted to understand this a little bit better, and we used a technique called sort-seq, where you actually deposit an individual macrophage in a well, and you're able to actually sequence that particular well. By doing this, you can understand the diversity of the populations of macrophages that are being uncovered. So we're able to see M1-like and M2-like macrophages, and even a progenitor-like population. And when we look at the populations, you can see these particular ones are activated and inflamed, whereas this population is the one that's active for lipid metabolism, and it's also active in other things that make you think these are the populations that are eating. So we focused on this particular population of macrophages. Using this, we were able to find markers of the lysosome that were very strong in this particular population. And this is very consistent with it eating the macrophages. And there's even some genes like this heme oxygenase that's very specifically localizing the macrophages that are doing the eating. So again, these are very phagocytic cells that are really interested in pulling in cholesterol, but also interacting with those stem cells. Now, we've gone and looked at the stem cells that have been eaten by the macrophages, with and without the macrophage, I should say. And we don't see any transcriptional difference at all at the single cell level between the stem cells that are being eaten or not. And we think that what's going on is actually more of a cell biological event rather than a deterministic transcriptional event. So we're trying to understand what is the code that actually drives these stem cells and macrophages to interact. So we were able to collaborate with a laboratory over at Harvard that does proteomics on a very, very small number of cells. In this particular case, Sam actually cut off the tails of the fish because the niche is over here, and then deposited 120 RUNX1 positive cells, these are the stem cells, or the macrophages, into these wells and did mass spec. Now, what came up in the macrophages, which was quite remarkable, were a variety of surface proteins that interact with different cell populations. And in particular, our attention was drawn to calreticulin and calreticulin 3. These are two different types of calreticulin, which are known to be prophagocytic signals. So calreticulin is mostly an ER protein, and it has a KDEL sequence on it. The KDEL sequence can actually be cleaved off, and when that happens, calreticulin appears on the surface of cells. It can then bind to receptors that are on the surface of the cells and be displayed. And this can become a recognition sequence for an eat me signal. This work has been popularized by Irv Weissman, where you have things like CD47 and SERP alpha, which are don't eat me signals. But the eat me signal is actually a calreticulin that interacts directly with surface receptors that's actually a lipoprotein receptor, LRP1. And this kind of forms a scavenger type of receptor to deal with the eating of the stem cells. So we've been able to find an antibody to calreticulin that stains our sorted cells. And now we're trying to develop evidence that the calreticulin is required for the interactions. Well, one of the things that we found was that on the macrophages is the LRP1 receptor. So these are the cells that are going to be doing the eating. And so the calreticulin that is presumably on the surface of the stem cell is able to interact with those macrophages. The other thing we've done is to start knocking down individual calreticulins. And you can see here that if you knock out CalR, you get a small effect that is not significant. But if you knock out CalR3 and CalR3B, you get a more statistically significant effect with regard to the stem cells and how they engage with the macrophages. And now we're looking with our clonal markers to see, does this affect clonality? But it may be that these CalR3s are very important for this recognition code for the stem cells. So just a working model is we think that there's a lot of stress in having the stem cells being born, and they need to traffic to another place and undergo mitosis. And this allows the stem cells to have three different paths. In one path, really nothing happens because these stem cells don't express calreticulin, they don't interact with those macrophages, they just persist. If you have some calreticulin on the surface of the stem cells, it'll interact with those macrophages that have LRP1, and this will lead to that interaction, the grooming behavior. Ultimately, the stem cell that's groomed goes on to divide. If there's too much calreticulin on the cell, then these macrophages will fully engulf the stem cell and kill it. This happens about 80% of the time, the middle one, and the bottom one happens about 20% of the time. Now, why go through all this type of a recognition? Well, again, there's this inflammation that is happening at the endothelial level, and these cells have to migrate. And so this is some mechanism of determining the number of stem cells and the ability of the stem cells to actually go on to be successful as executing a stem cell function. And I think since there is a recognition system going on, it's possible to manipulate this system to actually look at clonality. So I'm just going to leave you with my last slide, which just discusses the barcoding strategies for pediatric cancer in general. We've already talked about in the center here of using cellular barcoding technologies to evaluate the origin of leukemia, particularly pediatric leukemia. The technology we're developing in the blood system can be used to study any individual pediatric tumor. You could think of, let's say, rhabdomyosarcoma. What are the cells that are actually important for rhabdomyosarcoma? Again, is there a pool of progenitors that are amplified? And this leads to ultimately a transformation event that occurs later in life, or is thinking about glioma. So this barcoding technology is going to be quite useful in determining when these events happen, what is the cell of origin, and then ultimately how to treat it. So I just want to thank the people who've done the work. I believe I named everybody here. I did talk about Sam's work and Julie Perlin. We were greatly helped by Alex Shear's lab and Aaron McKenna and Henry Feldman. We've assembled a team, Team Barcode, which is very exciting, with many of the barcoders throughout the world. And we're sharing our information and trying to understand more about the technology. And then again, I want to thank Alex's Lemonade Stand and the RUNX1 research program, as well as the Evans Foundation, who've been able to help with the funding for some of the work that I presented. And with that, I'm happy to take questions.
Wow, that was excellent. That was amazing. The questions are rolling in, so I'm going to just jump right in. If you're up for it, we're going to go as many questions as we can. Okay, first question is from Borhane. Could you comment why TP53 mutation is found so high in zebrafish clonal hematopoiesis compared to mammalian counterparts?
Yeah, I think that, you know, we obviously the system we used, we actually engineered right from the beginning to put these populations in. It's likely with the P53 mutations, which has been examined in irradiation and chemotherapy, to be selected for. It's possible that because we're going through embryogenesis, there's a lot of stress on the system. So P53 clones end up getting selected a little bit more than what happens in aging, for instance. So I think that's likely the mechanism.
Okay, the next question comes from Maggie. And the question is, have you correlated clone lineage biases with malignant potential or development of disease? For instance, is an unbiased clone, which you may hypothesize is closer to a stem cell, more aggressive than a clone that matches a lineage?
Yes, so this is exactly what we'd like to do. So we are going to start to engineer some of the pediatric leukemia mutations into this model. And we will then evaluate what are the signatures of gene expression that predict aggressiveness, and aggressiveness in a loose term, you know, what predicts clonal expansion early, what predicts transformation into a leukemia later. And those are some really important things that we're just beginning to start to do.
Okay, the next question comes from Diane Krause. What is the likelihood that Gestalt will label a stem cell in progeny pair? What % would be missed due to efficiency of labeling and depth of sequencing? On the other hand, were there Y cell clones in the thymus that did have a matching HSC?
Yes, so there were clones that had a matching HSC, and a lot of clones had matching HSCs. So thank you, Diane, for mentioning this. We're labeling at a very high efficiency with the UB fish. We label about 17% of all of the cells. I think this can even be improved as we have some new tricks up our sleeves with getting UB clones, promoters that express very widely. So I think we won't have any trouble detecting stem cell clones. We can see the stem cell divisions. So I think that there won't be a problem with that. And again, the thymus clones are both ones that are represented with the marrow and others that are completely independent.
Okay, the next question is from Gulsi Person. First, great presentation. And then it says, I was wondering if these unbiased myeloid and lymphoid biased stem cells are distributed homogeneously or occupying distinct niches in the fish kidney.
Yeah, that is a really good question also. So it's really hard to do a sampling spatially with the Gestalt method, but with the Brainbow method, we are trying to do that. Cindy Dunbar had a beautiful paper several years ago showing spatial localization of individual clones. When we've looked just grossly at our kidney marrows, they seem that most of the clones are pretty well distributed, but I think we need to do a better job at looking at the microscopy and to be able to see if we can see subdomains within the compartments. The stem cells seem to be adjacent to venous sinusoids, so I think there's opportunities for these subdomains, but we would probably need to use the color system to do that rather than the Gestalt system.
All right, the questions are flying in, so I'm not sure how much time you got, but we're going to push through until you say you can't do any more.
I'm totally fine. Yeah, absolutely.
Okay, so the next question comes from Tim Treesh. Dr. Zahn, has your group applied something like, I'm not sure of this word, Cassiopeia to the generated lineage tracing data?
Yeah, just doing it now. So you could tell in my future direction slide for that Gestalt is we have the lineage tracing, and Aaron McKenna is really helping with doing this technology. And he has a few programs that he's actually working on. So those lineage trees that you saw in that one diagram were actually real. They did come from our data, but we're just getting comfortable with that data right now.
How'd I do on that word?
Perfect.
Oh. All right, well, we have Linda Resar in the house, and Linda says, great talk, Len. Would you expect the CALR mutations found in MPN would disrupt the macrophage HSC interaction?
Yeah, we've been discussing this. So, what happens is you have CALR mutations that lead to a myeloproliferative syndrome. And this is due to the CALR actually, the calreticulin directly interacting with the thrombopoietin receptor and driving that particular lineage. So we've wondered if the proliferative event that we're seeing after being partially eating a stem cell is directed, maybe not by thrombopoietin, but by other growth factors that are sensitive to this calreticulin method and in generating that proliferative division. We've actually labeled the cells with Fucci, which allows you to tell the phase of the cell cycle for which the division is happening. And I think a growth factor signaling mechanism would be very attractive. But we haven't put in the CALR mutation yet. It would be a nice thing to do.
Okay, Seth Corey says, very nice work. And how do you think absence of osteoblast and stromal cells in kidney perivascular area affects clonal outgrowth in adult hematopoiesis, and whether BM osteoblasts or osteoclasts might serve for macrophages?
Yeah, I think that's great. So thanks, Seth. So essentially, I just met with Chloe Barron this morning by Zoom, who's the postdoc who's doing some of this work, and she's been labeling the niche with the Gestalt system. And I think that's going to be pretty exciting, both stromal cells as well as endothelial cells, and then measuring how that will affect clonality. As many of you may know, there are mutations that can occur in the stromal cells that are pretty interesting that also seem to stimulate myelodysplasia or myeloproliferative syndromes. So these are some future experiments that we're testing, but no data right now.
Okay, the next question comes from Zubo, and the question is, are the macrophages eating specific HSPCs?
We think it's pretty generic. I'm favoring a model right now where the level of calreticulin on the surface is actually what's being sensed. But there are opportunities, even because there's really 2 or 3 calreticulins that get presented. So there is an opportunity where one of the calreticulins has a different function than another one. But I think the simplest model right now is just that the level is actually being determined, but it's generic in a mechanism in terms of this cell interaction.
I can't get the number of new messages to go down. It's just these questions keep flying in. Can you overexpress CALR on the HSCs to see if they induce a favorable selective clone? Comes in from Rosandra Kaplan.
Yes, and that was brought up earlier, and yes, we would like to do that experiment. It'd be a good experiment to do. We also would like to put in other oncogenes. We've been overexpressing some Hox genes and other things that might drive stem cell development, so that'll be pretty interesting also.
Okay, the next person, James Pallas, says, great talk. Anna Rita Migliaccio showed macrophages interacting with erythroblasts that then divide. Might there be parallels between macrophages, HSC, and macrophage erythroblast interactions?
Yep, I think Anna Rita had a very, very nice story on TGF-beta also, and looking at interaction of stromal cell, stem cell interactions. I think that if you remember, one of the markers we saw was heme oxygenase for these macrophages. And that particular marker, that gene, its function is to degrade hemoglobin. So maybe that's the macrophage that's eating the red cells as they senesce. So there might be some parallels. There are some erythroblasts there, but to be fair, again, we had done this experiment where we looked at the interaction of the macrophages with red cells versus stem cells, and the stem cell interaction was much more potent. So I don't know if it's CALR that's being detected there or some other mechanism.
Okay, you have one that says fantastic presentation, and then we have a question from Yashashwini. Could it be possible that grooming the macrophages might have an effect on the bias of a stem cell towards a certain blood cell population?
Yes, absolutely. So, you know, what happens is you have the birth of the blood stem cells, and then they have a certain diversity. And Elaine Dergiak had shown, for instance, that BMP was one of the signals that could allow for certain stem cells to have certain effects later in life. So I think this grooming behavior could have a functional consequence. And again, something we need to look at.
Well, Kelly Bailey said, thank you for an amazing presentation. And that is the last of the questions. We made it through.
Wow.
Amazing. So with that, I will wrap up. Len, we really appreciate it. And thank you for your time, and keep up the great work. And I know that you look forward to getting back to the lab full time.
Yeah, well, it's great. We're at 50% right now, and we're really looking forward to getting back to 100%. So thanks. And I hope everybody stays healthy during this particular time.
Thank you. We appreciate it, and keep up the great work.
Thanks.
All right. Bye-bye.